Hello, welcome to a GHGA webinar. Today we have with us Stephan Ossowski, and the talk title is Rare Disease Diagnostics in Complex Genomic Regions with Long Reads. And now I would like to introduce GHGA for those that are new, joining us for the first time and maybe don't know who we are. We are the German Human Genome-Phenome Archive, and we are providing a platform for human omic data sharing in a GDPR compliant manner. That means we make available omics data, clinical omics data for research, and we are doing in a responsible and legal and fair way. And we aim to contribute to biological discovery and to translation of research in science in genomic medicine. That's leads good with the topic of the talk today. And of course, we cannot do it together. We are a lot of and diverse audiences involved in that. And that's the reason we are organizing events like a webinar, or we have also lecture series. And this month, on 15 October, we have a symposium on site in Heidelberg in Marcelius College, but also you can join us virtually. We have a lot of great speakers. And if you would like to join our lecture series or our next webinar next month, we will have our ethics experts, Andreas Barms, and you can learn more about genetic discrimination. So you can visit our page. I will post it also in the chat, or you can sign up also for our newsletters to follow up. And now I want to introduce our speaker today. It's a pleasure for me to having here Stephan Ossowski. He makes his PhD in next generation sequences methods for plant genome analysis at the Max Planck Institute for Developmental Biology. After that, he worked as a group leader at the I have the name in Spain, I need to translate it, at the Central Regulation Genomic in Barcelona. And during this time, he started working and to explore with read technologies. And nowadays, he is leader of the Genomic Informatics Group at the University of Tübingen. And he and his group develop next generation sequences analysis methods and clinical decision support system for rare disease and cancer diagnostic. And today he will talk us a little bit more in detail about these jobs. So thank you very much, Stephan, for being here. And I hand over to you.
Right. I hope you can see my full screen now. If not, please let me know. And I shortened my talk title a bit just to Rare Disease Diagnostics with Long Leads is wrong. It should be long reads, obviously. So that happens if you shorten your talk title before the talk. I'm sorry for that. It's long reads. Okay, little disclaimer. I have shown very similar slides in recent talks, one for ESHG and one for the talk series of the sequencing centers of the NGS-CN. And if you have seen them, I'm very sorry. The last 4 slides or so will be new for you. The rest is a bit of a replication. Okay, so let's have a look at diagnostics in general. Because this is GHGA, I will focus even more on explaining bioinformatics, less wet lab part, but obviously you have the wet lab part as a start. In rare disease, you typically have index cases, the blood samples, or you might have parent-child trios, maybe even larger families with multiple affected patients. But in general, you collect the samples and you sequence, and from there on, bioinformatics basically starts. And you have to solve 2 separate issues. One is the basic analysis of your data, which typically starts from storing the raw files, down to some sort of prioritization of causal variants with a lot of stuff in between. Everyone knows alignment, variant prediction, annotation, and so on. And then a separate issue is the interpretation or helping clinicians and geneticists to interpret the data to identify causal variants, and these are called clinical decision support systems. And nowadays, they often try to include AI, but most of the time they are just fancy filtering software to allow people to reduce the number of variants that are possible solutions to the problem. Now, for nanopore reads, we have in both sections, we have to adjust our short read software. Looking first at the basic NGS analysis pipelines, you now have these nanopore long reads, which beside our normal analysis using alignment to a reference genome, which we have followed for a long time for the short reads for basically 20 years now, you can now think about other options like genome de novo assembly. All of you have likely seen these papers on telomere to telomere assemblies, where you can now get better assemblies than the actual reference genome. And then you would consider whole genome alignments, possibly of multiple full genomes. And nanopore, as well as the other long read technology, PacBio, allows you to read the methylation in the same run. So you can now also look at CpG methylation, which can be a cause of rare diseases and is even more interesting for cancer diagnostics. Finally, there's another thing you can do now with long reads, which is haplotype phasing. So you can phase the variants to know which variants occur on the same chromosome and which occur in the other alleles. So you have your two alleles phased, which can be very interesting, for instance, in recessive disease, if you have to make sure that the two variants are on the different alleles. So lots of interesting things. I have a little example here how that can actually impact the later step, the interpretation step. Normally, we look at our reads in IGV to check if everything is correct, the variant is there. But if you actually haplotype phase your reads into an allele, or you even do de novo assembly, then you don't have all that read stacks anymore. You can look in, that's an IGV picture, where you simply have two alleles now for each person you sequenced. In that case here, a family with two daughters, father and mother. And you can then see these inheritance patterns where the daughters inherited two insertions, which were heterozygous in father and mother, which could then be causal. In that example, they are not, but it looked so nice, so I chose that one. So you can represent large families very easily in one view, and much better than having all these big read stacks as in the past. All right, so for that reason, we had to adjust our variant analysis pipeline. We developed for a long time already a pipeline called MaxZap for medical genome sequence analysis pipeline. And that pipeline we had to adjust now basically from the mapping, or even before that, from the quality control to the mapping to all the different variant prediction tools for small variants, structure variants, CNVs, and repeat expansions. To continue, for instance, Minimap is now used for alignment instead of BWA-MEM. You have other QC tools available. You have other variant callers like Claire3 and Deep Variant often use neural networks to better interpret the pretty noisy squiggle data from and base calls from nanopore. You also have different structure variant tools. CNV calling, actually we use the same as for short reads because we develop it ourselves. And you now can also do stuff that was much harder with short reads, like repeat expansion detection, or also analysis of very repetitive regions, or regions that at least with short reads are repetitive. There's other tools I cannot show here. It's just not enough space, for instance, for the haplotype phasing, for the methylation analysis, for long read RNA, and so on. It's quite a big pipeline, but pretty much every step had to be replaced. Oh, in fact, here that's the phasing, haplotype phasing tool. You can go to our GitHub and check out our tool, Medical Genome Sequence Analysis Pipeline, but you can also go to other very good pipelines like Oxford Nanopore Technology has their own pipeline, Workflow Human Variation, very good and uses very similar tools as we do. Most of them overlap. Or you can go to NVIDIA, they have Parabricks. It's not as complete as these two pipelines, but they offer some cool speed ups for GPU, for instance, for Minimap and for deep variant, which you can also integrate in your own pipelines. So I list them here mainly because they develop the models for the speed up on GPU. There are, I'm sure, other pipelines in Nextflow and so on that you can find if you Google. The next step would be a clinical decision support system. Again, this is the one from us here called GSVAR for genome sequence variation. It looks like a fancy Excel or not so fancy Excel, depending if you like our graphical style or not. But you have for each variant an entry here. You have a large filter section. You have a lot of link outs to clinical databases and genomic databases for additional information. And then you have all the different types of variants and also RNA and liquid biopsy up here. All of these had to be adjusted for Nanopore, as I said before, and I will show in later slides. There are a lot of changes with Nanopore, starting from the quality of the calls and then going also to the amount of specific calls. For instance, much more structure variants you will have than compared to Illumina short reads. And this had to be adjusted. So our tool offers, for instance, the selection of specific phenotypes that will reduce the number of possible genes that will be shown that can be causal. The copy number and SVs and so on are there. Repeat expansions we had to update a lot because with Nanopore you can much better call repeat expansions than before. You will get more than twice as many structure variants, correct structure variants compared to Illumina at a much better precision. You can also do long-read RNA-seq, which will allow you to even better identify transcripts. And if you have splice variants in rare disease, these are very helpful. And as I said, specific for cancer, we have other options like liquid biopsy. And the final step will always be then a clinical report. In a research study, you will not have these, but if you work in a diagnostic setting, you would want to generate a clinical report that then lists the most important information for the clinician. In rare disease, that would be a causal variant. In cancer, that can be multiple somatic mutations in oncogenes and tumor suppressor genes, or also fusions and so on. So to set this up, our goal was to get accreditation for Nanopore sequencing for diagnostics. We already had that. It's similar to a TÜV for cars. You get accreditation for laboratory diagnostic tests, or also called LDT, laboratory developed tests, from the DAX in Germany, in the US from FDA and so on. And in that sense, you have to basically follow specific SOPs for every step in this long street from sample to final report to the clinician. Again, I will focus on bioinformatics mostly today, where we have the analysis software, the databases, and the graphical user interface for the geneticists or clinicians. And specifically, I will focus on quality control in the next few slides, and then more on the variant types after that. To establish that, we didn't want to go alone here in Tübingen, and we founded a consortium together with other university clinics in Germany. The green ones here were the initial founders, Tübingen, Aachen, Hannover, Berlin, and now several others have joined. And we have associated sequencing centers, and now we even have we just started a European version led by Olaf Ries. called LRIN, which will do the same Europe-wide, basically consortia supporting the introduction of long reads, on specifically nanopore reads in diagnostics. I will stick to the German one for now because there we have actual data in a round-robin test. So it's a German initiative. We were starting with 4 centers. We chose well-characterized patients for a cohort of up to 1,000 samples with a pilot study of 80. And we added the reference samples from Genome in a Bottle to compare the centers to each other. And our aim was to demonstrate that this actually helps patients. There is an added diagnostic value. We have a proof of sustainability in clinical practice. That is mainly the questions, can we actually do that? So question 1 is can we sequence thousands of genomes? And question 2, can the bioinformatics store all that data and analyze all that data efficiently? And finally, we have to do benchmarking and accreditation, which includes benchmarking of bioinformatics software on reference data. So all these SOPs, for us, the most interesting here are the bioinformatics SOPs. As I said before, for benchmarking, Genome in a Bottle is especially useful. I will say something for people who have never heard of that, although I think most people know what this is. And then we had real patient samples in the pilot included, but in total we want to go for 1,000 samples. If you are also a wet lab person, that's the current flow cell and pore and chemistry called R10.4.14 with V14 chemistry. We are currently testing the next generation, which has an improved motor protein and more yield, which I unfortunately don't know the numbering of the latest one. And then we have clinical use cases to test. And here we focused on stuff that is better with long reads than with short reads, which are repeat expansions or generally any type of low complexity or repetitive regions. Also including genes that have a pseudogene copy. So again, they are repetitive because of their gene copy. There are pathogenic haplotypes like the obscene cluster. Again, these are clusters of genes which are highly similar, but depending on which types you have in your cluster and which variants, you might have color blindness, for instance, in that case. And imprinting disorders, these are specifically for methylation disorders, so methylation-based diseases. First of all, let's get to quality control. That's the first thing we do, and it mostly falls to the bioinformatics to give a first overview of how good a run has been. One important thing is coverage or yield per flow cell, because we run 1 flow cell per human genome. And then we see quite a range of yield we get from like 100 GB up to sometimes 180 GB. And for diagnostics, we really like to get 40x coverage. I will show later why, but both for short and for long reads, this gives you significantly improved quality compared to 30x, which is the typical threshold for research projects. So if you get 100 GB out of your flow cell, it's fine, it's usable. If you get 120 GB out, it's much better for diagnostics. This is mainly a problem of the flow cells and how many pores are usable. Pores over time, they clog, they get stuck with DNA inside, and then you see this decline of useful pores. And then there are some tricks. You can flush the flow cells, you can top up with new DNA, and this way you can extend the lifetime of your pores. But at some point, your flow cell will be unusable, roughly after 3 days. But some flow cells have a very bad behavior. They clog very fast, their pores, and they are basically beyond saving. And then you get some of these outlier flow cells, and often nanopore replaces them. So you can see an overview of our pilot samples. As I said, in total there were 80, but because we are 4 sites, each site did 20 of them. And then you can look at some of the most important quality criteria. One is the N50 length of the reads. That's exactly defined like in a genome assembly quality. So it's you can see it as an average read length. It's not exactly average, but it's close to that. And we normally target 20 kb. In this test here, we had a few outliers because we used DNA from older samples, which was not as fit anymore and was already used for short reads. But if we have fresh and high quality blood samples, we would rather go for 20 kb. So it was not fully optimal, the test. Then you can see here, as I said before, how many gigabases output you have, which gives you the average read depth. We reached about 35, but we had some of these outliers here, which were lower. And that's very important for our so-called gap statistics. If we have less than 20x at a position, then we consider it not anymore useful for diagnostics. And the lower your average coverage, the more positions you have that are not covered with 20x. So with a high coverage, 99.8 positions have 20x. But if you have these bad outliers here, suddenly more than 25% of positions drop below that 20x and become gaps for us. And then you have to redo the sample or fill the gaps with other technologies. Also interesting for you might be runtime, computational runtime. It's actually fairly doable on a normal server. However, we now achieve much better runtimes using GPU servers and big NVIDIA servers with their H100 chips. You can bring that down to 2 hours instead of 20 to 30. So that slide is a bit old. Now coming to the benchmarking, as I mentioned, Genome in a Bottle is what we use as reference. They have multiple families, especially a big one from Utah, including this famous NA12878 sample, or also called HG001, but also some other ethnicities and some other families. We chose HG001 and 002 for the simple reason that HG001 is best annotated for single nucleotide variants and indels, and the number 2 sample has the best structure variant analysis. And in Genome in a Bottle, what they did, they used a lot of sequencing technologies to basically get you the absolute gold standard. And then they divided it in the high confidence region, where they are really 100% sure, and then the lower confidence regions. These are repetitive regions where Illumina, for instance, has a very hard time with the short reads. So to create that, they included pretty much all known sequencing technologies and even some non-sequencing technologies like bio-nano optical mapping. And by integrating all of that, they pretty much, well, get close to a golden set of correct SNVs, indels, SVs, copy number variants, and so on. And that's what we take then as a benchmark. We can do that because you can order these samples. So you can really test yourself from wet lab to bioinformatics using the same samples. There will be some people telling you that when you order these samples, there will be slight differences. There might be one extra SNP because of whatever damage, or when they multiply the cells, they divide the cells, there could be additional mutations. But compared to the 4 million SNPs in the germline genome, I think that's negligible. So don't let people confuse you. These are very good. Order these cell lines and you can do all your accreditation and benchmarking on these cell lines. Looking at that, we can now measure and compare different tools. For instance, here a comparison of Claire 3 and the old version of deep variant called Pepper Margin deep variant, the new one, deep variant 1.5, I think, doesn't need this Pepper Margin anymore. But that is not that important now. More important is that you can see that at about 30x coverage, both tools achieve close to 100% precision for SNF calling. Of basically all talks I'm asked, is that real? I heard nanopore is the biggest crap with a lot of errors. That's completely wrong. At 30x coverage, your consensus will not be full of errors. You will have a very high precision. Why people still believe that is probably because of the indel issue. Everyone has probably heard that indels are not that great with nanopore because their most common error are gap errors. So a base is missed or sometimes added, especially in homopolymers. And you can then see that in the precision, which never, even at very high coverage, can cross the 95% threshold. And there's also a big difference between the tools. Here in precision for indels, Claire 3 is significantly better. And you can also see here why we like high coverage. You can still gain a lot going beyond 20x up to 60 in precision. And these are the two samples we have done. You see the same for SNVs for recall. At 20 to 30x, you are at 100%. So absolutely no problem there. You can do SNF diagnostics without a doubt. It's as good as short reads. For the indels, it now swaps. Deep variant has better recall, worse precision, as you can see here. Now, deep variant is better in that sense. I have to say, we have not found the perfect indel model yet, and there is still work to do. We here, in that case, have a recall of only slightly above 80% in whole genomes. To make that a bit more specific, we divided whole genome and protein coding because a lot of diagnostics happens on protein coding exons. In whole genome, I just showed you the graphs. SNFs is close to 100% precision recall and genotype accuracy, but indels have mainly a sensitivity problem, but also reduced precision. Genotyping is fairly okay. But this improves a lot in coding, and the simple reason for these roughly 14% improvement is that you have much less homopolymers in coding, and these are the main problem of nanopore. So this is results with the Dorado 5-base model. 5-base model means, or 5 or 6 base, both exist, that they also call methylation at the same time. I will show that later. And here is very quickly an analysis of how much coverage you need. For SNVs, you are saturating basically at 20x. That's our gap threshold, where we say below that, we consider the gap. At 30x to 40x, there's almost no improvement anymore. I mean, it's minimal. Different for indels, you get an improvement with every 10x extra, and it continues actually even for higher coverage. So there is still gain from more coverage. Even if you have that coverage, we are not happy with that. I mean, 94 and 97% recall and precision is not what we want for diagnostics. We want to have it better. So we investigated different ways of getting better data. One is that we are currently testing this new flow cell version and new chemistry with new motor proteins, and improved pores can also lead to improved base call quality. But one option that ONT is now investigating are the duplex reads. And duplex reads means that you simply sequence the forward and then the reverse strand. And because you have then both, you can find out in which positions they disagree. And these you can then mask by making it an N, for instance. Or you can make a more involved algorithm correcting then and choosing the correct base in the two strands. And you can see here that improvements to the pore can be extremely massive from R9 pore, which I think was switched to the new R10 pore, it's about a year ago or maybe even 2. That was a massive improvement. But if you now go from this current chemistry and you do duplex, you have almost no errors left anymore. You get a very clean alignment in IGV. So improvement in the chemistry and pores and novel ideas can combine to be a huge improvement. We looked at the so-called simplex reads. Here are, you only get the forward read sequenced, and then the duplex reads. Here you get forward and reverse read sequenced. And we saw some interesting effects. For instance, similar to short reads, even long reads slowly lose quality over their length. So at the end, they drop in quality slightly, not as much as Illumina reads, which would already drop after 100 base pairs. But even long reads do that. If you make duplex, because you sequence from both ends, you can correct then, and you get an equal quality mostly across the whole length. And you get, by that, you then get a much higher Q30 percentage. So bases that have at least Q30 quality. And that is then also visual in your variant or in your error rates, especially interesting for us in the error rates of insertions or deletions. So bases that are wrongly inserted or wrongly deleted, they drop about 4-fold to 5-fold. And similarly, mismatch rates, where you simply have a nucleotide, a wrong nucleotide change, also it is reduced by 70%, so 2/3, which is also quite strong. And we also see less soft clipping, so more parts of the reads are aligned, which could be either because the quality is good to the end, but I just learned recently that it's also because in the duplex algorithm, the algorithm removes adapters automatically. So part of that high soft clipping rate of simplex is probably because we don't do adapter clipping. We let Minimap do the clipping for us, and that is then looking like a soft clip. So we are working on that now. We are checking if we do adapter trimming, if actually the difference becomes smaller because between simplex and duplex. The third option you have to improve your quality are machine learning based methods that try to fix the quality of the reads. So they run before you do anything, before you do variant calling. And one interesting example is Hero or Hero. I don't know exactly how to pronounce it, which is a haplotype-aware error correction and was published recently, I think, as medRxiv or bioRxiv. And I assume it's kind of a consensus type where you correct reads that you align to each other, and haplotype aware simply means that you have longer stretches where you correct errors, assuming that you only have 2 haplotypes possible. And if you see more than 2 in these reads, then these must be sequencing errors. So we are also working on that, but based on the publication, they claim that they go from Q20 to Q40 on average, which would be huge. That's Illumina level or even better quality. Now let's come quickly to the variants. A lot of variants improve using structure, using nanopore. The most obvious is structure variants. Structure variants are quite long, and with short reads, you have some options. You have this paired end mapping algorithm, PEM algorithms. You have split read or clip read algorithms. Like here you see these clipped reads, which are only half aligned. But all of them are, in the end, statistics that often give you wrong results, but especially with short reads, you miss about half of the structure variants. Complex ones, very long ones, there's different reasons. Also, very short structure variants are often missed by paired end mapping methods. With nanopore, that's very different because you get the actual alignment across your whole structure variant. You can consider that always as a split read in a sense when you compare to short reads, but where you have much longer anchors on both sides of the structure variant. And here's a comparison to the physical mapping bio-nano. Looking at that, we compared again on genome in a bottle sniffles and QtSV, two well-known structure variant callers. And what we could see is that at different coverage levels, here you see how much coverage we used, 5x, 10x, 15x, 20x. Their recall and precision goes, once you reach 30x, basically goes close to 95. Not exactly 95, but 93%. For sniffles, even if you use low coverage, it performs pretty well. QtSV has a big drop at low coverage, but is slightly better at super high coverage. We decided to go on with sniffles because it's very robust to non-perfect coverage. And then if you look at the subsampling experiment again, where we try with 10x, 20x, and so on from the same sample, you can see that 30 to 40x here's again the golden spot where you get best results. So that's sufficient for sniffles. If you have super high coverage, you could switch to QtSV because it's mildly better, but I think it's not worth having to maintain two software in your pipeline. If you compare that to short reads, I don't show it here, but I can tell you Illumina reaches about 45% recall in that same genome in a bottle. So you get much more, but also Illumina doesn't reach precision over 90%. Ivo Goodone said they have 50% recall, of which half is wrong, and that is short read structure variants. In long reads, you have now about 23,000 structure variants across the whole genome, and most of them are actually correct. That's very different because now you have to look at much more structure variants than short reads. And while in short reads, you often removed a lot of your candidates just because in IGV you saw they are wrong, that will not happen so often anymore here. So what are other things we had to look at because they improve? I already mentioned because of haplotype phasing, we can better interpret compound heterozygote variants in recessive diseases. So if you have a gene and each allele in your index patient is affected by a different position, then you have, you are sick. And for that, you have to make sure that these variants are really on different alleles, not on the same allele, because then you would still have a healthy allele. And that has been done in the past by trios. You check that one came from the mother, one from the father. But if you can do haplotype phasing correctly, then you save two sequencing experiments. I just mentioned structure variants, that they are now twice as good, and that includes also mobile elements. And now we will have a quick look at duplicate genes, repeat expansions, and DNA methylation. Duplicate gene is any gene, like here SMN1 in spinal muscular atrophy, where there are other gene copies in the genome which are highly similar. In SMN1, it's even more complicated because SMN2 is a bit shorter, but there can be multiple copies of it. And there can be longer and shorter copies of it. So it's quite a mess. And with short reads, they normally just cross-map between all the copies. And then if you actually had in the important SMN1 gene a deletion, like here a homozygous deletion, you couldn't see it because the reads from SMN2 just cross-mapped in here. And you missed the deletion. But with long reads, they can be correctly assigned to the other copies, and then you see your large deletion clearly. So that's huge because there are more than 200 clinically relevant genes that have such pseudogene copy. That's just one disease out of at least 200. PacBio released a cool tool for that, and it covers 160 of them. So we can now do diagnostics in 160 genes we couldn't do before once we have it fully implemented, and that tool is called Paraphase. Next, repeat expansions. These are motifs in the genome which are already repetitive, so-called simple tandem repeats, but which can, in a patient, suddenly have a huge expansion. So there are each green thing here is one motif copy, and they have much more copies of the motif. And using the long reads, we can not only correctly align them, but we can phase each read to its allele, and then we see the short allele and the long allele nicely. And with that, we can then generate consensus repeat length. You can also do that in histograms. Like here, the short one is about 55 or so copies, and then the long one is much longer. That's the idea. There's also a tool for that called Straggler. In fact, there are multiple other tools like tandem genotypes and repeat HMM. Master student in my group, Thomas Brown, compared them, and we found that Straggler is superior to the other tools. With one exception, it has no graphical output, so we simply added all the graphical output ourselves and made an update called Straggler on as an add-on, so that you can get fancy pictures. Here are some examples. Huntington's disease, people have heard of that, I'm sure. And in IGV, you see here the long allele, the expanded, and the short allele, which is not expanded. So the long one basically looks like an insertion of 53 CAG repeats in the gene HTT. And that's causal, and you can segregate it in the family and so on. You can also simply look at the histograms. These are the reads from the short haplotype, and these are the reads of the long one. You can see they are not all equally long. That is both a technical and a biological reason. Technically, because of the long repeat, even nanopore has some slipping, probably it misjudges the exact length. But also biologically, because when you replicate the DNA during cell division and so on, there are actual changes. So not even all cells in the same person have the same repeat length. So there is some repeat length variability. Here's another example in ataxia. Another case we had in our pilot study. And again, you see the big difference here. You can also see it in IGV. It's a 77 CAG repeats in ataxin 3 and solves the case. It gets more complicated the longer your repeat expansion is. Here we have already more than 300 extra repeats. And because that's a motif, you have up to 1,000 base pair longer insertion. And you see that here it's still quite well visible in IGV and was also predicted by Straggler. And our final example was not any more predictable by Straggler. It has more than 10 kb repeat expansion of a much more complicated hexanucleotide repeat. And you can see here that only 2 reads managed to span it. If you would have sequenced much deeper, maybe we would have gotten more reads. But what you can see is that in most cases, Minimap simply gave up and clipped the reads of these red things here. So we have to look now for other tools that are not just based on the reads that align across the repeat expansion, but also on read clipping in the vicinity to get a better prediction. So this C9orf72 in ALS was not detected by Straggler. Finally, I mentioned methylation calling. Very important, we now for every nucleotide in our read, we not only have the nucleotide and its quality, but also the methylation, if it's a C in a CPG context. And that can be important in some regions, so-called imprinting loci, where you inherit a highly methylated and a highly unmethylated copy from mother and father. And if that goes wrong, and you, for instance, have both copies highly methylated, you might have a gene that is shut off because the promoter is methylated, or that is overregulated, so overexpressing. And this can be easily detected by simply plotting the methylation of the two alleles. Similar to the variants, we can phase the two alleles. And then Methyl Artist is a nice tool to show you this as a plot, and everyone can immediately see here everything is fine. We have a methylated and unmethylated copy in that imprinting locus. Here is one example where things go wrong. Here, that's a control. There are some methylated and some unmethylated reads shown in red and blue. And in the case, we have only unmethylated. And the reason here is that there was simply a deletion of the methylated allele. So this should have also been detected by a deletion caller, but short read it can be unreliable, as I said before. So it was missed in the past potentially. With nanopore, you should now see a large deletion and a methylation change. So you have double proof of your event. And that's a case of autism spectrum disorder with the gene SNRPN. I'm not sure how many people are interested in the actual disease genes here, but bioinformatically, we can solve that now. And there's other examples which now combine the last two topics, repeat expansions and methylation, because many repeat expansions, like here, one in FMR1, an X chromosomal gene, when this repeat expansion gets at least 200 copies long, then the locus is hypermethylated, and then the disease is different and stronger. So that means having the repeat expansion alone with, let's say, 100 extra copies can give you some sort of mild phenotype, and having it very long plus the methylation, hypermethylation, gives you suddenly an even stronger phenotype. I'm not a clinical doctor, so if you want to understand that exactly, there are good papers discussing this Fragile X syndrome. But again, bioinformatically or even just visually, we can see that difference now in nanopore. So one final story for that, you can also use it not just to identify known causes of disease, but we also used nanopore and PacBio sequencing to find a completely new repeat expansion locus in the gene ZFHX3, encoding a polyglycine, so the amino acid based on the GGC codon. That was a case which was long time unsolved, more than 25 years. Our collaborator from Utah has this huge family, which they have long-term records of all family members because they are Mormons and they record all their family history over centuries. They were originally from Sweden and moved to Utah about 200 years ago, I think, or 150. And we had families which were like up to 6 generations apart, and we still knew it, and all of them had the same phenotype. So you would think that should be super easy to solve. All of them should have exactly the same causal variant. Now let's just find it. But it wasn't found for 25 years. They even had a linkage region. They knew it must be on one arm of chromosome 16. But that was as close as they got. And so we fired with long reads 6 cases of this family. We chose from each branch and 2 controls, 2 healthy family members. And then we used PacBio and ONT to sequence them. And lo and behold, yes, we found this locus, this GGC repeat, simple tandem repeat, which in healthy people was just 20 copies. These are the 2 controls here. And in all other family members we sequenced, they had a short allele and a long allele. And the long allele was at least 42 or longer. So that was the shortest one here. So that seems to be the shortest pathogenic one. So that means if you have 42 repeats, you have the ataxia disease that this family has now for a long time. We could validate that here, for instance, with another repeat expansion tool from PacBio called Target, TRGT, very good tool, makes very nice plots. And you can always see the short and the long allele, short and long, short and long. You can validate it using PCR amplification. Here you see the long one. Here you see the controls. They don't have it. And we also looked in our large database of, I don't know, 11,000 or so genomes. And all the ones over 40, except for one, have this ataxia, spinocerebellar ataxia 4. This one, we couldn't figure out. It's a breast cancer case, and maybe it was a false prediction because it was short reads, and then you have to use expansion hunter, and expansion hunter is not so great. So what does that mean? Normally you have 20 copies and then you have 20 glycines in your protein. But if you have the expansion, you suddenly have a much longer chain of glycines. And there are multiple explanations why they can cause a problem. Either they just break the gene itself. But what is more common in these polyglycine and polyglutamines is that they are toxic because these long chains here aggregate in cells, and over time, and that's why many of these diseases are late onset, it gets worse and worse, the accumulation of these in the cells, and especially in these long-lived neuronal cells. We went on because we had this super cool family. We said, let's look at the haplotypes and see how they share haplotypes across the family. We took the best assembled person and defined the disease haplotype by just saying all SNPs that are on the same haplotype as the repeat expansion is 1. And all SNPs that don't match that must be the other allele. You have 2 copies of every chromosome. So then we can check in the other family. So that's family 3, first person. Here's family 2, first person. And you can see, yes, it had exactly this haplotype. Almost the megabase was shared between these 2. And with a repeat expansion. So they really have them from the same ancestor. In family 1, we also see that haplotype here in red, but there was a crossover at some point. So now you have it starting on one allele and then continuing on the other. Here is the repeat. And with all that information, we then checked in our database for more. And we were lucky to discuss that case with Malte Spielmann from Lübeck, and he had also a case where he was just solving it and it turned out to be the same. And it turned out that these people from Lübeck are coming from the same ancestor. So they are basically long-term ancestry from one single original case. They share the same repeat expansion and the same haplotype from Sweden. When you look at these SNPs in the haplotype, we looked closer and we found 7 SNPs that are very, very close to the repeat expansion. And you only find them with the repeat expansion, not in the other alleles. So we said, let's use them to screen our short-read genome databases to find more cases. In short reads, repeat expansion prediction is hard, but if you find these marker SNPs, then you could assume they also have the repeat expansion. And we did that and found, in fact, quite a lot. So as I said, there were 2 from Lübeck in addition. There were now 7 from Tübingen. And Tübingen is not fully correct. These patients actually come from different cities in Germany. I remember there were one family from Magdeburg, which were just sequenced in Tübingen. So they don't live here. And what we found is that they share most of these marker SNPs, and we could validate all of them, except that one of the SNPs doesn't seem to be in the southern or middle German samples, but it's in Utah and Lübeck. And we could see that this SNP is completely absent in GNOMAD, and it was likely happening after the repeat expansion. So there was a branching, and we can even distinguish now, because the family is so old, that one branch of the family had a novel SNP that the other branch doesn't have, but they share the other 6. So with that, we by now have more than 20 people identified also outside of Utah that actually have that. The latest stuff we do is to use nanopore instead of for rare disease, for cancer. And one of the most impressive things I've seen published in recent years was the sequencing of tumors during surgery. So during the surgery, you have about 2 hours to sequence your tumor, classify what type of tumor it is, and that might lead to an adjustment of surgery during the surgery, because maybe in a specific subtype you have to cut out more, and in another subtype which is less aggressive you cut out less. And that's only possible with nanopore. We joined the Leopard consortium led by Philipp Euskirchen for that. Only difference we make is instead of the Minion, which most people use, we will use the Promethean, but that doesn't make a bit of difference. Philipp Euskirchen, he developed these NanoDX pipeline, and especially the classifier cross-NN, which can classify tumor or cancer subtypes, and is based on low-pass nanopore whole genome sequencing. And there are two neural networks pre-trained. One is just for brain tumors and one is for all types of cancers. With that, we got in a round robin test some samples sent from them without telling us what they are. And then you have to go through this quick analysis. You have a very short DNA QC, library construction 15 minutes, and then you sequence up to 6 hours, but you don't have to, because NanoDX or cross-CNN updates every minute in real time what it currently thinks it sees based on the methylation profile of the DNA. And you can see here that in our run, we basically knew already after 5 minutes that it's most likely that blue one. And I'm sorry, we didn't replace the numbers yet by the name. It's a meningioma. You could wait for 20 minutes and then you are like 90% sure. And that's all the other options that are there, other brain cancers. You can see there are almost 0 chance. And NanoDX is nice. It makes you a report in the end that includes its prediction. It's a bit small here, but this is really reading meningioma, and it's all basically 100%. It shows you all the other cancers it tested, and it also gives you, from the low coverage, copy number profile. So that's really great. You can do that really in 1 hour during surgery. We are now adjusting that also to liquid biopsy and other ideas. So I think this is one of the most powerful new technologies I have seen in recent years. With that, I want to thank all the people from Longer, Elrin, and especially my institute and our collaborators, ONT and NVIDIA, the Longer Consortium, and that's still the posters from ESHG, I'm really sorry. And the experts from Tübingen. If you have questions, let me know. We have several people here.
I want to say thank you, Stephan. Thank you also our audiences for the time and the interest. And thank you our GHGA colleagues who helped to set up the webinar also. And yeah, see you in the next webinar. Have a nice day.
Nice day, and thank you for joining. Bye-bye.
Bye.